Hi, my name is Daphne Superville. I'm a third-year PhD candidate in the Biomedical Sciences program at UCSF, and I'm doing my thesis work in the lab of Dr. Melissa Reeves, where I'm focusing on understanding the role of tumor heterogeneity in squamous skin cancer metastasis. Tumor heterogeneity is a term used to describe the genetic diversity that we see among cells within a tumor. Although most tumors begin from a single mutant cell, over time, cells will acquire additional mutations, leading to a tumor that's comprised of several genetically distinct but related subpopulations termed subclones. In the Reeves lab, I'm using a carcinogen-induced mouse model combined with an in vivo fluorescence-based lineage tracing system to study tumor heterogeneity. In this model, subpopulations within malignant tumors are labeled with different fluorescent proteins, and when these tumors metastasize, we find that multiple subpopulations from the primary tumor are able to see these distant metastases. This has led me to investigate how different tumor cell subpopulations may be interacting with each other to promote metastasis. My experiments so far have focused on a cell line derived from a multicolored polyclonal lymph node metastasis, which we call 1241 LND. This cell line has a CFP-expressing subpopulation and an RFP-expressing subpopulation. By isolating these subpopulations and re-injecting them into mice, I've been able to investigate how these cells behave and metastasize differently when injected together or individually. What I found in these experiments is that individually, the RFP population is a much more efficient metastasizer than the CFP population. But when injected together, the RFP population does not metastasize as efficiently. As you can see in this graph, 50% of mice injected with only RFP cells came down with metastases, whereas the various other mixed conditions all exhibit fewer metastases. I also analyzed the immune infiltrate of these tumors by flow cytometry, as a growing body of work is beginning to show an important role for the immune system in tumor progression and metastasis. I found that certain immune cell populations, macrophages and neutrophils in particular, were differentially abundant in RFP-only versus mixed conditions, indicating that the presence of CFP cells may alter the immune microenvironment. I'm currently conducting follow-up studies to confirm whether this CFP subpopulation is actually suppressing metastasis of the RFP cells, and I'm also beginning to further characterize the immune infiltrate differences we see between these two populations. But my hope is that this work will shed light on how the diversity that we see within tumors contributes to their malignancy and how tumor cells may be interacting to facilitate metastasis. Lastly, I'd like to give a big thank you to the ARCS Foundation and its members for supporting this work, as well as my PI, Melissa, and my co-mentor, Dr. Andre Goga, for their guidance and mentorship. Thank you.